Good day, everyone. I am Padernal, together with my partner Silmaro. We'll be discuss the topic entitled Inheritance of Quantitative Characters Multiples Gene. Okay, for definition of quantitative characters, it is an inherited character that expressed phenotypically in all degrees of variation between one often indefinite extreme and another. Quantitative character. Quantitative character is another name of quantitative trait. This refers to species characteristics that can be expressed numerically. So when we say the word numerically, it is interpreted through numbers. These phenotypes expressed often fall along a wide spectrum of variance. Okay, to explain genotypes of an organism is its complete sets of genetic material. Genotype can also be used to refer to the alleles or variants an individual carries in a particular gene or genetic location. So if you still remember the discussions, if both alleles are the same, the genotype is referred to as called homozygous. So in other words, it is a combination of two dominant alleles. So if the alleles are different, the genotype is referred to as heterozygous. So heterozygous is a combination of one dominant and one recessive alleles. The genotypes contributes to phenotype. So the degree to which genotype and phenotype depends on the trait. For example, the petal color in a pea plant is exclusively determined by genotype. The petals can be purple or white depending on the alleles present in the pea plant. There are three available genotypes: homozygous dominant, heterozygous, and homozygous recessive. All three have different genotypes, but the first two have the same phenotype, or purple, as distinct from the third, or white. In 1903, Wilhelm Ludwig Johansen, or he is the best known for coining the terms gene, phenotype, and genotype, and for his pure line of beans or Bacillus vulgaris. So beans, he used the beans or Bacillus vulgaris in experiments in genetics because the beans has a unique characteristics.
Good morning, sir. By the way, I'm Romel Silmaro. So my next topic would be to be discussed is multiple genes and the inheritance of the character. Suppose we know that a gene individually producing a slight effect on the phenotype, but in a combination with a few or many other genes controls as a quantitative character is called as a polygene. So since there are many genes of this kind for a quantitative character, they are also known as a multiple factor, each factor having too small an effect to be traced. So those genes or alleles will contribute to the trait involved are called contributing effective or active genes or the alleles. So those alleles which do not contribute to the trait are called non-contributing or non-effective or non-genes. So take note that those do not contribute trait are the non-genes or the alleles. So this is George Ornioli. So George Ornioli is a British mathematician. So according to him, in 1903, suggested that a quantitative characters are controlled by many genes or cumulative genes or the multiple factors of or a polygenes that is termed coined by matter. So this is this is Kenneth Mather. So, according to Kenneth Mather, polygenes are not allelic of each gene has a small or a similar effect. Genes are additive or cumulative. So, the net effect on the other trait will depend upon the number of the contributing alleles or effective alleles present that is called a multiple factor of hypothesis. So, in the Punnett square, the F1 heterozygous or the hybrids in all cruces showed a lighter red, kernel color, so or known as an intermediate color. So then the homozygous zygous or the red kernel colored parents. So he got different F2 phenotypic ratios and various hybridizations such as 3 red is to 1 white, 15 red is to 1 white, and 63 red and is to 1 white. So take note that as always we have one white color because of the homozygous recessive. So moreover, there was a continuous variation in the red kernel color to the white kernel color in F2 genotypic ratios in the generation. So let us consider a cross between a red kernel color with variety with a white kernel colored, where the two genes, each gene has two alleles are being involved. So let the homozygous AEBB has a four contributing alleles or the effective alleles, be the genotype of the red kernel colored with variety, and the homozygous recessive, the white kernel colored, has only non-contributing alleles. So the genotype of the F1 hybrids will be heterozygous, or the two contributing alleles, since the F1 hybrids have only two pigment contributing alleles. The kernel we have only the color which is the medium red colors, or known as the intermediate color. So in the F1 hybrids with the genotype heterozygous, produce only four types of gametes such as homozygous AB, heterozygous AB, um, and heterozygous recessive and homozygous recessive. So that is what happened during the gametogenesis. So the random fusion of the gametes will result in 16 possible combination of 4 by 4 in the F2 regeneration. So out of this, 15 will have red, and 15 of will have red kernel color in the contributing alleles, and we have also one color depending on the number of the contributing alleles, and host effects are similar and additive, and one will be the white kernels. So that is 15 red is to 1 white. We're going to show the figure that was given in the Punnett square. So if we're going to look the red kernel color wet times white kernel color wet, where the two genes are being involved rather. So if we're going to look in the first column in the first column of the homozygous parent, there is a four alleles, four contributing alleles, and the color is deep red, or the grandparent color. So since in the second column we have the heterozygous AB, so the result is there is a three alleles, and the color is dark red. So in the third column, we have three contributing alleles, and the color is dark red also. So in the fourth column, we have the recessive, and we have the heterozygous recessive, and we have two alleles, and the color is medium red. So in the next second column, in the second rows, we have the three contributing alleles, and the color is dark red. And the second is we have two contributing alleles, and the color is medium red. And the third is same, we have two contributing alleles, and the color is medium red. And the last, we have one contributing alleles, and the color is light red. So in the third column, in the third rows, we have three contributing alleles, and the color is dark red. And in the second, we have two contributing alleles, and the color is light red, medium red. And in the third, we have the two contributing alleles, the same as we have the color medium red, and lastly we have the one alleles and the color is light red. So in the last column, in the last rows, we have two alleles and the color is medium red, and we have one contributing alleles and the color is light red, and last is, and we have also the one contributing alleles and the color is light red. So in the last column, in the last rows, there is no alleles, which is the color is white. So among the F2 progeny, we have 1 over 64 has 6 contributing or effective alleles and will be like the red grandparent color. So second is we have 6 over 64 and has 5 contributing effective alleles and the color is red colors. So in the third, we have 15 over 64 and has 5 contributing alleles and the color is red color. So in the fourth, we have 20 over 64 and has 3 contributing alleles and the color has medium red color. So fifth, we have 15 over 64 and has 2 contributing alleles and the color is light red. So in the sixth, we have 6 over 64 and has 1 contributing alleles and the color is very light red. And lastly, we have 1 over 64 and has no contributing alleles and the color is white kernel. So that was a color of the combination between the red wet variety and white variety. So thus there was 7 phenotypic classes will show in a continuation variation such as 1 deep red is to 6 dark red is to 15 red is to 20 medium red is to 15 light red is to 6 very light red is to 1 white. So in the ratio given is 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. So kernel color in wet being a quantitative trait is also modified by environmental factors such as temperatures. So which changes enzyme control reaction rates, sunlight and nutrition or the etc. So which change the color of the level of enzymes and substrates for reaction. So such environmental effects cause individual grains of weight to vary within each genotype.